Good morning to all. Hello friends. I am your friend Dr. Shubh Patel. Today we will discuss about a question from Lab Animal Medicine. Which of the following nomenclature designations listed below refers to a consomic inbred mouse strain? A C57BL/6J.129/JDB3J. MRL facile pair. C57BL/6JYDOM. CRLNMRI. DBA/homeo 5ad-plus. Answer, CC57BL/6JYDOM. First, what is a consomic strain? A consomic strain, also called chromosome substitution strain, is an inbred strain in which one entire chromosome from a donor strain is introgressed into the genetic background of a host strain. Example: C57BL/6JCHRYDOM, where chromosome Y from the DOM strain has been introduced into the C57BL/6J background. Now, let's go option by option. Correct answer, C, C57BL/6JYDOM. Breakdown. C57BL/6J is the host strain background, C57BL/6, Jackson Lab substrain. YDOM means the Y chromosome from the DOM donor strain. This is the perfect example of consomic nomenclature. Host strain plus chromosome, Y, plus donor strain designation. Justification: this is a classic consomic inbred mouse model. Only the Y chromosome from the DOM strain is introduced into the C57BL/6J background. Usage example: researchers use consomic strains like this to study Y chromosome effects on fertility, aggression, metabolism, etc., while keeping the rest of the genome constant. Incorrect answers: A. C57BL/6J.129/JDB3J. Breakdown. C57BL/6J.129/J suggests a mixed background, B6 and 129 strain. DB3J likely refers to a specific mutation, DB gene, diabetes, from Jackson Lab. This is an example of a congenic strain or a hybrid, where a specific mutation is transferred, but it is not a whole chromosome substitution. Why incorrect? This is more typical for congenic nomenclature, mutation introgression, not consomic, whole chromosome replacement. Example: C57BL/6JDB/DB mice carry the leptin receptor mutation and are used to study obesity, not a consomic model. B. MRL facile pair. Breakdown. MRL is the host strain. Facile pair refers to a mutation in the FOS gene, lymphoproliferation. This is a spontaneous mutant strain, typically used for autoimmunity research, lupus models. Why incorrect? Mutation in a specific gene, not a chromosome substitution. Not a consomic strain. Example: MRL/LPR mice develop systemic lupus erythematosus-like disease. D. CRLNMRI. Breakdown. CRL stands for Charles River Laboratories. NMRI is a popular outbred stock, derived from Swiss mice. This is an outbred stock, used for general-purpose experiments. Why incorrect? Outbred stocks are genetically heterogeneous. Consomic strains are inbred with targeted chromosome substitution. Example: NMRI mice are commonly used in toxicology studies due to their robust nature. E. DBA/homeo 5ad-plus. Breakdown. DBA/HA is an inbred strain, DBA strain from Harwell. Myo 5ad-plus refers to a specific allele of the myosin VA gene. This indicates a mutation in a gene, not a full chromosome introgression. Why incorrect? This is a mutant strain, not a consomic strain. Example: DBA/2J strain is used in hearing loss research, but not in consomic studies. Summary table. Final note: when reading mouse nomenclature, consomic strains will typically have host strain, CHR chromosome donor strain, or host strain-chromosome/segment donor. Example: C57BL/6JCHR1PWD/PH rightward arrow chromosome 1 from PWD/PH strain into B6 background. This is useful in mapping genetic traits to specific chromosomes. More examples of consomic strains. C57BL/6JCHR17PWD/PH. Entire chromosome 17 from PWD/PH strain into B6 background. Used to study immune system genes, MHC complexes on chromosome 17. C57BL/6JCHRXMSM/MS. X chromosome from MSM/MS strain into B6 background. Useful for studying sex-linked traits, behavior, and metabolic syndromes. C57BL/6JCHR13A/J. Chromosome 13 from A/J strain into B6 background. A/J strain is susceptible to lung tumorigenesis, so this consomic model helps map tumor susceptibility loci. Pattern: background strain plus CHR plus chromosome plus donor strain. Quick cheat sheet: how to read mouse strain names. Tip: when you see CHR, think consomic. When you see specific gene mutations or supplier labels, it's likely not consomic. Where consomic models are critically used. 1. Hypertension research. Example: Shore CHR13BN strain. Hypertensive rats with chromosome 13 from normotensive BN strain to identify blood pressure control genes. 2. Type 2 diabetes and obesity. Example: C57BL/6JCHR17NSY. NSY mice are diabetes prone, substituting chromosomes helps identify diabetes susceptibility loci. 3. Autoimmune diseases. Example: B6CHR17NZB. NZB strain is lupus prone. Chromosome 17 carries immune-related loci. 4. Behavior and neuroscience. Example: C57BL/6JCHRXMSM. To explore X-linked genes affecting behavior or cognition. 5. Male fertility. Example: C57BL/6JYDOM. Male fertility and hybrid sterility models to map Y-linked traits. Bonus tip: how they make consomic mice, breeding strategy. Start with 2 inbred strains, recipient, example C57BL/6J, and donor, example DOM. Cross them and back-cross the offspring to the recipient strain. At each generation, select offspring carrying the donor chromosome. Continue until greater than 10 generations equals 99.9% of recipient genome, except for the substituted chromosome. This ensures stable background. Only the donor chromosome influences phenotype. Conclusion: exam-ready statement. Consomic strains are powerful tools in genetics where one chromosome from a donor strain is introgressed into the inbred background of a recipient strain. This enables precise mapping of traits associated with specific chromosomes while minimizing background genetic noise. Thank you for listening. Have a nice day ahead and save your patient with love and dedication. Please subscribe and like. See you soon on next video. Till then, bye from Dr. Shubh Patel.